Okay, so it's a great pleasure to have Professor Giovanni Berlucchi. Professor Berlucchi doesn't need an introduction. He's Professor Emeritus at the University of Verona. And it's really, we're talking about more than 50 years of great research. He moved from anatomy to electrophysiology to neuroimaging. Now you can really see through this presentation that he had really trained with great names. So the talk, Giovanni, he's an expert on visual spatial functions and vision in particular, but also I think at heart he has the corpus callosum. But what I asked him here really to expand a bit on the history of neuroscience as well. And it'll be a great pleasure to have someone really who was there in the exciting times to tell us about the life of Moruzzi and the people that really contributed to the history of neurophysiology in the last 50 years. Thank you very much, Giovanni.
Thanks, Marco. I'm very grateful for the invitation and for the honor and pleasure to speak here about my teacher in neurophysiology, a man who has practically founded neurophysiology as a discipline by itself in Italy, and who had very deep roots in this country. You know that neurophysiology has two British fathers. One is Charles Sherrington, who was professor in several places, Liverpool and eventually Oxford, and Adrian, a real giant, Lord Adrian, who was the first to demonstrate that neurons talk to each other and with the body with action potential, that the action potential, the frequency and distribution in time of action potential is the communication code of the neuronal systems. And Moruzzi has a very interesting initiation to science, because in a sense he is the descendant of 3 Nobel Prizes. When he was a medical student in Parma, a very young man, 20 years or so, he worked in histology with this man, Antonio Pensa, who actually, when he was probably my age or older, which means 83 or 84, gave me the histology examination in Pavia. This man also trained the young Moruzzi in the early 1930s in histology, and this man was direct descendant of Camillo Golgi, the Nobel Prize. 1906, was professor of pathology in Pavia. But he also was the descendant. Pensa in 1930, when Moruzzi had done his thesis with Pensa, his thesis was on the cerebellum. Actually, you know, the cerebellar cortex was a battlefield between Golgi and Cajal, and the basket cell synapses on the Purkinje neurons, according to Cajal, were the demonstration that there is a gap between the presynaptic and the postsynaptic neuron. But Golgi said, no, you're wrong, because he said that there was a small fiber leaving from the presynaptic terminal of the basket cell and going into the granular layer. And the first Moruzzi's work was on this topic. But he was also descendant of Charles Sherrington, who got the Nobel Prize in 1932, because these two men trained him before he became a professor of physiology. One was Mario Camis. Pensa left Parma in 1930 when Moruzzi was 20. He asked Moruzzi to follow him in Pavia, but Moruzzi he didn't like anatomy so much. And he preferred to go into physiology. And his professor of physiology in Parma was this man, a very interesting man, Mario Camis. I will say something about him, who had been a pupil of Sherrington in the early 20th century, in 1909, when Sherrington was in Liverpool. And also Moruzzi worked in Brussels with Frederick Bremer, who was also a pupil of Sherrington. And then the other Nobel Prize, British Nobel Prize for neurophysiology, was Lord Adrian, the future Lord Adrian. And Moruzzi worked directly with Adrian in 1939 with a great, I think, a great paper, a great discovery. So there are 3 Nobel Prizes as the predecessors of Moruzzi. Camis was a very interesting man who had a tragic life. He was from Verona, the city where I live now. And he was of Jewish descent. He was very interested in physiology, and he worked with great Italian physiologists, including Luciani. And then he moved to Pisa, but he didn't like working in Pisa, and he went to England to work with great physiologists, first in Cambridge with Langley and Barcroft. Actually, he took part with Barcroft, he was interested in the dissociation of oxygen from hemoglobin, and they made an expedition to Mount Rosa to see how exercise could influence. Actually, they discovered the fact that salt, organic salt in the red cell, can influence the dissociation of oxygen from hemoglobin. Camis had a tragic life because his wife died of abdominal typhus in 1930, and he converted to Catholicism. Actually, he became a Dominican friar, and then he took the orders. But nevertheless, because of Mussolini racial laws, he was thrown out of the university when he was professor in Bologna after Parma. And even more tragically, when he was readmitted to the academy in Italy, he was thrown out again because he had been rector of the University of Parma, and he had written something in praise of Mussolini. So he had 2 expulsions. And he could not be rehabilitated because he died in 1946. He was a very interesting, you can see, as a rector of the University of Parma and as a Dominican friar. And even he officiated at Moruzzi's wedding, I don't think very well because he was inexperienced. And he made a very, in Sherrington's laboratory, he made a very important discovery, which Sherrington then named occlusion. Occlusion is the phenomenon by which, if there are 2 pathways converging on a center, on a nervous center, you cannot get a summation of the 2 responses because they occlude each other. So the total response is less than the sum of the 2 inputs. He didn't use, Camis didn't choose the word occlusion, but was a very good paper. Sherrington was very fond of this paper. But he was also a man of wide, broad culture. And he wrote about emotions. He sided with Sherrington. Sherrington was against James' Lange idea that emotions are due to the reverberation of bodily inputs on the brain. The psychic content of emotion is due to the body, in a sense. And he also wrote a very important book. This is still, it was written in Italian in 1928 and translated into England by Creed, who was also a pupil of Sherrington. As you see, the book was dedicated to Sherrington. It is still cited as a reference book. If you look up the usual Google Scholar and so, is still cited as the basic reference test on the vestibular apparatus. And you can see a letter that in 1934, Mario Camis wrote to Sherrington, and the letter says that he had a very young student, Moruzzi, who was working on a subject of interest to Sherrington. Sherrington had shown that if you cut the spinal cord, there are some changes, histological changes in the motor neurons, which he attributed to deafferentation of the neurons, and he called transneuronic degeneration. And Moruzzi's thesis in physiology was done on the Purkinje cell in the cerebellum after cutting the spinal cerebellum, and he found the same thing. And Camis tells Sherrington, we are working on your lines of research. He wanted to say, I am still your pupil, your follower. And you see, Dr. Moruzzi is a very promising young fellow. Moruzzi was 24. And I cherish the idea that he could someday come to spend some time at Oxford. With kind regards from you, for Lady Sherrington, your affectionately Mario Camis. Here is Sherrington, and as it turned out, Moruzzi didn't go to Oxford, but he went to Cambridge to work with Adrian. You can see the years later, and they together, they did, this is probably historically the first registration of single action potential of cortical neurons in mammals. They stuck an electrode in the decussation of the pyramids, microelectrode, and stimulated or acted on the motor cortex of the cat. And these are the first action potential recorded in 1939 of single cortical neurons. To my knowledge, this is the first paper on the activity of cortical neurons and single. They put strychnine in the cortex, they stimulated electrically the cortex, but most of all, they showed that even in anesthesia, when the cat was not moving, there was continuous discharge from the cortex, in confirmation of the idea of Berger that with the EEG, that the cortex, even in anesthesia, the cortex is active. And you can see Moruzzi wrote about it. And then, but the most important discovery of Moruzzi was done with Magoun, who was then at Northwestern University in Chicago. And Magoun here in 1961 was with Bremer, who was another. Moruzzi and Magoun did this paper which changed the history of neuroscience by demonstrating that if you stimulate the reticular formation in a slightly anesthetized cat, you can produce an arousal reaction in a much easier manner than if you stimulate the sensory pathways. And then Magoun and his co-workers demonstrated that lesion of the reticular formation produce the synchronization of the EEG and typical coma. And you can see that in humans, also in humans, in a relatively recent paper about 12 years ago or so, Damasio and Parvisi demonstrated that the neuroanatomical correlates of brainstem coma are lesion in the reticular formation. And Moruzzi, in the 1950s, he proposed this idea that I would not see any objection to the hypothesis that this central tone in the activity of the cortex might represent the neurophysiological correlate of states called wakefulness, awareness, or alertness. He was wary of using the word consciousness because, as you know, in the crazy, the wacky behaviorist period, we were, as a young man, I was forbidden to use the word consciousness until, I think, 1975 or so. The study of the effect of reticular stimulation made it. Also, he had this idea, which unfortunately he did not develop, that the reticular formation was not, as Magoun thought, a monolithic system, but rather a connection of centers with somewhat all with arousal properties, but with somewhat different functional properties. In this paper, he had this idea, and it, as you know now, this is the old Magoun idea of the arousing reticular ascending system, of a system which from the reticular formation acts on the thalamus and the cortex. And this today is, of course, has been changed, and the idea is that there are several nuclei which acts on the cortex and use different transmitters. For example, the blue, the pedunculopontine nucleus, and the nucleus of minors are acetylcholinergic. The yellow is histaminergic, the raphe is serotonin, and the locus coeruleus is noradrenergic. And they are orchestrated by these proteins, the orexin, which are in the perifornical area of the hypothalamus. You can see here there are the different number of neurons. I won't go through this. It's too long. And the changes in the activity of these nuclei can produce what is known as the wake-sleep cycle, where sleep is divided into the two typical stages, one in which apparently there is no consciousness, no dreams, and the REM sleep in which there are ocular movements, but also dreams and some kind of consciousness. As a proof of what you can see, what an English psychologist in 1989 wrote in the International Dictionary of Psychology. Consciousness is a fascinating but elusive phenomenon. It is impossible to specify what it is, what it does, or why it evolved. Nothing worth reading has been written about it. But if you look at the textbook, one of the most read textbook of neuroscience is the one by Kandel. And if you look at the first 4 editions, and if you look for the word consciousness in the analytic index, in 1981, 0. In 1985, 6. In 1991, 7. In 2032, and now in the latest edition, I think about 100. Conscious and consciousness have come back. I've come back also for clinical reasons. Clinical reasons because now there are many patients who seem to have lost consciousness because of many causes. There are many of them, many more than in the past, because in the past they were going to die quite quickly after. It could be a vascular accident, it could be a trauma, it could be an infection, it could be a poisoning, whatever. And they would die in a few days, but now with the pharmacological and medical development, many of them are kept alive. And you can see that the word coma is now reserved to a relatively low condition which changes rapidly and which is characterized absolutely by inactivity. The eyes are closed, the patient does not respond, and usually it's something which doesn't last very long. But then you can have an evolution, and you can have the vegetative state, which is in the definition of the clinician, although this can be discussed, someone prefers to call it unresponsive waking, because actually there is no evidence for the presence of consciousness, but there is a sleep-wake cycle demonstrated by the fact that at some time the eyes are open, the patient seems to look around, although there is no direction in looking. You cannot have catch the attention by moving, by presenting stimuli. So there is a sleep-wake cycle, but waking is an empty state. There is something, the brain is able to keep, in a sense, the body awake. There are some movement, non-directional movement, non-directed movement, but apparently no conscious, although there are doubts about that. Chronic coma is very, brain death, of course, is the end of the person. And I will talk about this locked-in syndrome. The locked-in syndrome is a syndrome which was known for a long time, is a condition in which the patient seems to be in a vegetative state, in the sense that there is sleep-wake cycle, no movement, apparently no communication. But if you can use this small movement that the patient can make, sometimes you can establish full communication. So the problem is consciousness without behavior. The locked-in syndrome is a syndrome characterized by consciousness almost without behavior. I will come back. The most famous case of locked-in syndrome is of Jean-Dominique Bauby, who was a French journalist. He was supposed to be in a vegetative state for several months, no consciousness at all. And eventually a nurse realized that he could move the eyes and move the eyelids a little bit. And then she tried, she said, if you hear me, I don't remember what precisely, but blink 3 times. And he blinked 3 times. And eventually they established a communication, and he could, by moving the eyes, give yes or no answers, and also indication. And eventually he wrote a book, The Diving Bell and the Butterfly, which recounts how it is to be in that condition. And now it is disappointing when they leave the television on with the idea that can be a good stimulation for recovery, that the wrong channel was left on rather than the football match that he wanted to see. But another, only the eyes say yes, and with the eyes you can do. And two of my countrymen, neuropsychologists, Kappa and Vignolo, in Milan, were the first to perform in-depth analysis, and they gave an IQ test to one of these patients, and the patient was found to be able even to have emotion. He was fully aware of the handicap, and if they can communicate, apparently they want to remain alive, to be able to continue their life, contrary to our sensation that it would be like being buried to be in that condition. This is a relatively recent case from this country. Barbara Wilson is a well-known researcher in neurorehabilitation. Do you think things have improved since the time of your injury? Yes, I feel healthier now. And if you read these, really, the patient, what are your hopes for the future? I hope I can live more independently. Is there anything you miss? This was a woman. I miss being able to jump out of bed in the morning. I don't miss doing the ironing. So assessing another Englishman who is now in Canada, in New London, Ontario, where he has a huge center for the study of consciousness from the point of view of the brain. And he has been the first, when he was in this country, he has been the first to try to speak to patients apparently in vegetative state, not locked-in syndrome, but apparently in vegetative state, absolutely no movement. And he knew if you study in people with an intact brain, with a healthy brain, two states of imagination. If you imagine to walk through your apartment, the brain activity is in blue. You can, is very different from the brain activity if you imagine exercising in a motor activity like playing tennis. And the two patterns of brain activity are completely different. You can see that the cortical activity is much more lateral when one imagines walking through the house than when imagining playing tennis. And it tried in subjects with no brain lesion, and you can dialogue just using these two. You make up a code. One thing means yes, the other thing means no. And in this famous paper now, of a few years ago, Owen and his co-workers, they tried this on patients apparently in a vegetative state and obtained some kind of communication. In other words, there are patients who cannot communicate with their body, but they can communicate through their brain activity. Of course, this is open to all. Imagine all the possible obstacles to the use of this technique. For example, the patient may be deaf. There may be a lesion in the auditory pathways. If there is no response, it doesn't mean that he is not conscious. You don't know what. But if there is a response, you can. Of course, it's not. Nobody can do the kappa and Vignolo. It is very difficult to give an IQ test. But usually this method would take forever. But certainly this casts some doubt on the diagnosis of the vegetative state as a state of complete unconsciousness. And as a matter of fact, the people in Liège and also here, when Magoun was here, have evaluated that the possibility of error is at least 40%. So it's a tragic condition. Just to talk about, again, our countrymen, many, many years ago in the EEG journal, these two men who are now dead, Carlo Loeb, who was a neurologist in Genoa, and Gianfranco Poggio, who worked with Mount Castle in Baltimore for many years, a well-known neurophysiologist, they say that a woman with a lesion in the pons, who they thought was completely unconscious, had an EEG was by and large normal. There was a beta rhythm, even some gamma, and they said that the EEG is not, probably it's not. This, I think, Moruzzi was stimulated by this discovery to do an experiment with his collaborators. And if you consider the gray column as the arousing system, it's been subdivided in many things. We know that if you divide the brain from the brainstem at this level, at the level of the midbrain, there is coma, or certainly something like the vegetative states. If you make the lesion here, between the encephalon and the spinal cord, there is a normal sleep-waking cycle, probably with consciousness in the cat brain. But if you make the lesion here in front of the fifth nerve and at the midpoint of the pons, there is a very curious condition. If you make this lesion here, these two lesions produce abolition of arousal, but these lesions leaves the cat in a very strange condition. The electroencephalogram is cats, as you know, the normal cat sleeps most of the time. 65–9% of the time, a normal cat. On the contrary, a cat with the lesion at the midpoints is awake during 78% of the time. It can move the eyes only in the vertical plane because the brain is as the third nerve. The fourth nerve and the sixth nerve are either the lesion or behind the lesions, so the brain cannot move the eyes laterally. It can only, the third nerve can only move the eyes vertically. And if you move something in front of the cat follows the moving hand. If you repeat the stimulus, there is habituation. If you have a dog, if the dog barks, there will be a pupil dilation. There is an accommodation in response to zooming stimulus. And the pupil dilatation can be conditioned according to a Pavlovian paradigm, but I'm most interested in this last thing, the operant conditioning of the eye movement. And this was done, unfortunately, these two papers, which are old now, they were done by a Japanese neurophysiologist who was in Pisa in the 60s, and they learned to do the midpontine pretrigeminal preparation. And it used the method of operant conditioning by the self-stimulation methods. If you take a normal cat or rat or whatever animals, there are sites in the brain which, when stimulated, are behaviorally pleasant for the animal in the sense that the animal likes to be stimulated. If you give the animal the way to self-stimulate, if you give him the button, the animal on the liver, the animal will continuously self-stimulate. One of these areas is the lateral hypothalamus. Now we know that this is where the dopamine pathway goes up to the nucleus accumbens. The nucleus accumbens is probably what they call a pleasure center. Joseph Ledoux doesn't like this. He now thinks that you have to divide psychology from physiology. But nevertheless, there is no doubt that an animal likes being stimulated in that area. On the contrary, there are other points in the brain, for example, the deep layer of the superior colliculus, where stimulation is painful, disagreeable to the animal, and the animal will work to avoid. If you give him the button, he will never press it, but if you give him means to avoid the pressing, he will activate them. And Kawamura he asked himself whether the midpontine cat would use ocular movements to self-stimulate. So he had an electrode in the lateral hypothalamic area and gave the animal the possibility to self-stimulate itself by moving the eyes. And the result was clear. You can see when he gave the animal the possibility to stimulate, there was an increase in the frequency of eye movements, and especially if the animal was in the light. In other words, if the animal could see something. In other words, the animal used, like the people in the locked-in syndrome, he used eye movements to obtain something. I don't know if this can be called communication, but certainly it can be called a desire of the animal. I'm not talking about the feeling, because people like Joseph Ledoux say that we cannot talk about what the animal feels. But anyway, the interesting thing is the following, that if you gave the animal the possibility to interrupt stimulation in a region of the brain which appear dislikable to normal cats, the animal didn't care. It didn't do anything. And this is, of course, also Kawamura demonstrated that it was a real connection between if he disconnected in time the occurrence of the electrical stimulus and the eye movement, there was no conditioning at all. The animal had to realize, in a sense, that the present stimulus was associated with the stimulation. And this is a long story, but the conclusion of Kawamura, which is in a sense also congruent with a suggestion by Damasio. Damasio has suggested, Damasio in his books on feelings and the relation between the body, and Damasio believes that people with the locked-in syndrome in a sense have a shift of the balance between positive and negative emotion toward positive emotion. In other words, they are less sensitive to negative emotion, and this is because he calls this the brain-body loop. He thinks that emotions, like in the old ideas of even Darwin and James, of course, and Lange, that in order to be emotional, you have to have brain activity, of course, but also you have to have signals from the body. In other words, the increase in our frequency in fear is part of the cause of this psychological emotion. And Damasio felt that negative emotions are less, negative emotion require the body, the brain-body loop, whereas positive emotion can be realized, implemented totally within the brain. And the results of Kawamura would seem to, you can see there, this is the body. For negative emotion, you have to have a correspondence. For positive emotion, there is a loop which is entirely confined within the brain. These are there is an interesting finding. You know, people believe, people try to resurrect the James idea of emotion. If you ask some people to use the muscles of here, you know, if you use the corrugator, this occurs when you are unhappy. If you also contract the orbicularis oris, like here also is, although there is no felt emotion, and if you hold the pencil with the fingers, looks like a smile. And the idea is that if you have to recognize positive and negative emotions in photograph of faces, and you measure the accuracy of the response, if the contraction of the muscle correspond, for example, if you smile, the performance is a little better than if you frown. If the photographs are neutral, there is no influence of the smile or frown. And if you have to judge about, well, it doesn't matter. If you have to judge about an angry expression, again, if you frown, you're better than if you smile. This is the response to photograph of a group of people in locked-in syndrome studied by Roman Neapolitan colleagues. The paper was published in Journal of Neuroscience. And you can see that they have to perform on recognition of positive and negative emotion. And their performance is compared with that of people with intact brain. And you can see that the only difference is negative emotions. The people with locked-in syndrome perform less accurately than normal people on the negative emotion, which would be in agreement with both the cat experiment of Kawamura and the Damasio inference. But the body, of course, is very important. These are two, George Lakoff and Mark Johnson have, we were talking this morning about the body. The body, of course, is very important, and the proposal of these people, we think with our brain through functional neural circuitry, and thought is made meaningful by ways these neural circuits are connected with the body and characterize embodied experience. So-called abstract ideas are embodied in this way, as is language, mostly via conceptual metaphors. And the primary conceptual metaphors are affection and warmth. Warm means, there are experiments which indicate that if you shake hands with a person with a very cold hand, in a sense you are repellent. And morality, cleanliness, importance, weight, of course, aim, direction, happy is up, sad is down, more is up, less is down, a warm applause, a jelly handshake, and also the arrow of time. Past is behind the shoulders, and the future is ahead of us. And a recent experiment is very interesting in my mind, moving through time. If you have a person stand on a platform, or if you measure EMG, and you ask to think the person in one session, he has to think about what happened to him or her 4 years ago. And in another occasion, to imagine what will happen in 4 years' time. And look about the inclination. When one thinks about the future, during 15 seconds, there is a slight inclination forward. If you think about the past, there's a slight inclination backward. Let's finish going. Another experiment by the same group, you can have the sensation of being moved if you have the right visual stimulation. Remember, if you sit in the first row in a cinema, you may have vestibular sense, even nausea, because of this visual vacuum stimulation. And this was proved. What happens when you have a sensation of backward vacuum, you have to report, the mind is always wandering, and people are asked to report what they're thinking about. And you can see that during the backward stimulation, 63% were thought about the past, and much less thought about the future. The reverse when you have the forward action. In other words, sensation from the body influence your attitude toward the arrow of time, and in a sense are in accord with the idea that signals from the body can, of course, be generated by the brain, but in turn they can influence the activity of the brain. And this is Moruzzi, and I think I can end here. Thank you very much for the